Hey everyone, welcome back to my class. In today's class, we are going to study about the flagella. What is flagella? Flagella is basically whip-like structure. Here, if you see in the picture, this blue color one, it's going to give you the appearance of whip. Like a hunter hota hai na, that we call it as whip-like structure. It is thin, filamentous. Filamentous like this, like a tubular structure it will be, right? Microscopic. See class, flagella basically, if I'm going to take the example of bacteria, bacteria has flagella. Now on our palm we have n number of millions of bacteria, but are we able to see with our naked eyes? No, because they are microscopic, so we won't be able to see them, right? So if flagella is present on the bacteria, obviously we won't be able to see bacteria, so they are also microscopic. Flagella protrudes or extend from the surface of cell membrane. Put question mark over here, I'll be explaining you in the next slide. Number of flagella varies. Every organism have different number of different number of flagella. It could be one, two, or many. Is it present only in eukaryote or only in prokaryote? No, it is present in both eukaryote as well as in prokaryote. So if an organism is having flagella, that organism is going to be called as motile organism, and you will see movement in that particular organism. If flagella is absent in that organism, if any of the organism does not have flagella, so they are called as non-motile because they won't be able to move anywhere, right? Now see this structure. We all have studied the structure of cells. So this is, suppose this is a cell, and this is the cell membrane or plasma membrane, and then we have nucleus, and then inside we have cytoplasm. So here you can see class, this is an extension. Let me change the color. Okay, so here you can see class, here is the extension of plasma membrane. Basically this extension of plasma membrane is going to form your outer sheath of your flagella or cilia. Flagella or cilia. So there will be the extension of cell membrane, and this extension of cell membrane is going to form the outer sheath of flagella or cilia. Now inside this extension, what will happen? There is a basal body over here. This basal body is present. This basal body is nothing but it helps in the formation of flagella. Helps in the formation of flagella or cilia. So class, if basal body is not present, then there will be the formation of flagella or cilia? No, there won't be any formation of flagella and cilia. So basal body is the one who is responsible for the formation of cilia and flagella, right? From the basal body, this filament comes out. Basically it is going to form the main part of your flagella. Core of the flagella, we call it as filament or exonym. We call it as filament or exonym. Now see this sentence, the third one. Protrudes or extend from the surface. Flagella is the one which extends from the surface. So when we talk about eukaryote, it's the extension of cell membrane or plasma membrane. But when we talk about the prokaryotes, the outer layer is your cell wall. So there will be the extension of cell wall. I hope this is clear to you people. Now when we talk about the flagella will be different in prokaryote and in eukaryote. When we talk about the structure of flagella, basically the structure of flagella is there will be the basal body, then there will be a hook, and from the hook there will be filament. Okay, so this is your basal body, this is your hook, this is your filament or exonym. Class, please remember the name of exonym, super important. Okay, so when we talk about the bacteria, all the three components are present, but when we talk about the eukaryote, hook is absent. Hook is absent in eukaryote. Am I clear? Now, which protein is present in flagella of prokaryote? Flagellin protein is present. But what about eukaryotic flagella? Tubulin protein is present. Have to remember, very, very, very important. Now, these are the extension of cell wall, and this is an extension of cell membrane. It does not have 9+2 arrangement, and it has 9+2 arrangement. Hang on. Did I explain you about 9+2 arrangement? Cool. Put question mark over here, and we are going to study about 9+2 arrangement in upcoming slides. Okay, then we'll come back. Now, what are the components of flagella? I told you that it will be basically having basal body, then hook, and then your filament, right? But overall, if I want to talk about this exoneme wala part, this flagella, this filament wala part, which all components are present, that's what we are going to see. So the first one is your exoneme, second one is your microtubule, third one is your dynein, fourth one is your inner and outer sheath, fifth one is your radial spoke, sixth one is your basal granule, and seventh one is your lateral appendages. One by one, we are going to see each and every component of the flagella. Let's start with the exoneme. When we talk about the exoneme is that, just now I explained you, exoneme is the main part of the flagella, that is core. Core means again main. So it is the main internal structure. This is the extension of plasma membrane and this is the internal structure. So main internal structure is nothing but your exoneme. Okay, exoneme. It is the central longitudinal. How it is running, class? It is longitude. It is running in this way. It is present in the center and then running in this way. Is it running in this way? If this is running in this, if this axoneme is running in this way, we call it as transverse. But it is running in this way, we call it as longitudinal. We call it as longitudinal. Microtubular structure. So when we do the cross section of it, here you can see the cross section of it. So we will see microtubules. These are nothing but the microtubules. So that's what is being said over here, a central longitudinal microtubule. So it is present in the center. How is it? Longitudinal. And what is present? Microtubular structure is present in both cilium as well as in flagella. And it is arise from centriole. Just now I told you that the basal body is responsible for the formation of cilia and flagella. So in the basal body, basically your centrioles are present, which are responsible for the formation of flagella and cilia. It is surrounded by the membrane, which is continuous with the plasma membrane. What do you mean by this? Here you can see this is your exoneme. It is surrounded by the plasma membrane, which is continuous with the plasma membrane. It is surrounded by the membrane. This membrane we call it as outer sheath. This is in continuation with the plasma membrane, right? Now, all the component of exoneme are embedded in the matrix. What is matrix? I'm pretty much sure in 10th standard when you study about the blood, you must have studied about the components of blood. We have 4 components of blood, that is your RBC, WBC, platelets and your plasma. Now, if we study about the blood, we have studied that the plasma is the liquid component onto which your RBC, WBC and platelets will be found, right? So, it is a liquid component. So, this works as a matrix. Plasma works as a matrix. So, matrix is nothing but the liquid that fills the space. So, plasma is the one which is a liquid that fills the space of blood vessel, right? Microtubules run parallel to the axis. Put question mark over here. I'll be explaining it. Now, this structure of flagella is super important. Everybody has to draw this diagram. Okay. Now, it has 2 microtubules. What are microtubules? I'll be explaining in detail. First, understand the structure. Okay. Microtubules. Here we have 2 microtubules, that is A and B. A1 is complete over here. If you'll see the structure, A1 is complete but B1 is incomplete. So we have A microtubule and B microtubule. So can I say they are in the form of doublet? Yes, they are in the form of doublet, right? Now, obviously A is complete. So just remember that A is the powerful one. So if it is a powerful one, it will have the radial spoke. It will also have the dynein arms. Okay, dynein arms. Now, A is going to connect with the B1 of the other doublet through a protein called as nexin. What kind of protein it is? It is the elastic protein. It is the elastic protein, right? Now class, as I told you, A is complete, so A is powerful. A will have radial spoke which is going to connect with the inner sheath. Now in between class, if you will see, outer we have doublets. Outer we have doublets. These doublets are actually 9 in number. If you count 1, 2, 3, 4, 5, 6, 7, 8, 9. These are 9. Total 9 in number, right? Now the A1 is the one which is powerful. Just remember, like that powerful, I'm just randomly saying it to remember. Okay, because it is complete cycle. So it will have dynein arms, it will have radial spoke. These radial spokes are going to connect with the inner sheath. What one is your outer sheath? This plasma membrane is nothing but your outer sheath. Inner sheath is over here, which is going to cover your single cell, which is going to cover your your singlet. That means A microtubule. In central we have singlets, not doublets. Is A is accompanied by something else? No, it is single. So A is going to be not accompanied by something else. So we call it as singlets over here. These two singlets are going to be protected by the inner sheath. Protected by the inner sheath. Now this inner sheath are going to be in connection with the A microtubule. Okay, with the radial spoke. With the radial spoke. Again, these radial spokes are elastic in nature. What is the need for all these connection, ma'am? Why do we have radial sheath? They see. Basically, flagella is going to move like this. When flagella move like this, what happened? This movement of flagella is because of this sliding movement. You remember we have studied about the actin and myosin protein. So actin and myosin protein are going to slide over each other. Due to the sliding, contraction will occur, right? So same way, one doublet is going to slide over the other doublet, due to which there will be the movement in the flagella. So this movement, just imagine if it is not stick to somewhere else, what happened? There could be a possibility that this doublet will move and come over here. So this will be the disturbance in the structure. That's why we are having elastic protein. Why elastic protein? Because movement will be allowed when it is an elastic protein. If elasticity is not there, then what will happen? Whenever sliding occur, what will happen? There will be the rupture in the flagella. Okay, so I hope you understood the structure of flagella. Super important. We have two microtubules, that is A and B. A1 is complete, B1 is incomplete. A has radial spoke, which is going to be in connection with the inner sheath. A also has dynamo, which helps in the sliding movement of doublets, which helps in the locomotion of flagella, right? Now, so what is the arrangement over here, class? How many doublets we have? 9 doublets, where it is present at the periphery, right? And in the center, how many singlets we have? 2 singlets. So this is the arrangement of flagella. Now do you remember I told you about 9 plus 2. So 9 plus 2 arrangement is not present in prokaryote, but 9 plus 2 arrangement is present in eukaryote. So this was, I was talking about 9 plus 2 arrangement, right? Now, microtubules. Let's understand, ma'am. You told me about the microtubule A and microtubule B. But what are these microtubule A and B? First, let's understand about it. These microtubules are nothing but these are the network of protein filaments, which are essential component of cytoskeleton. Cyto means what, class? Cell. Skeleton means which gives us the structure. So cytoskeletons are the organelles which give structure to the cell, which gives structure to the cell. Different cells have different structure. So this structure is being provided by the cytoskeleton. इस cytoskeleton में क्या present होता है which helps in giving this structure? Microtubules are present. What are microtubules? They are the network of protein. Got my point? Now you will see arrangement of how microtubules are arranged in the flagella. You will see 9 plus 2 arrangement. It is made up of protein that we call it as alpha and beta. Okay, repeating myself again. So cytoskeleton organelle is present. Main component of cytoskeleton is going to be your microtubules. Microtubules will have protein, that is tubulin. Tubulin is of two types. One is alpha, other one is beta, right? Now here you can see, class, characters. If I'll talk about doublets, where we have seen doublets in the picture? Periphery. Singlet, where we have seen singlet? In the center. So number of doublets we have 9, number of singlets we have 2. So this gives the arrangement 9 plus 2 arrangement, right? Now here you can see very easily, class, I forgot to explain you over here. Now here, class, we have studied about alpha and beta tubulin. These alpha and beta tubulin are going to arrange in an alternate fashion to form one protofilament. Ma'am, are we supposed to remember this protofilament? No, you're not supposed to remember it. Just understand the concept. Alpha and beta are going to arrange alternatively to form one protofilament. Same way, if I want to talk about A microtubule and B microtubule. So in A microtubule, 13 protofilaments are there. This one is your one protofilament. You can see alpha and beta arrangement. Like this, you have 13 protofilament, and can you see this hollow? Can you see this hollowness in between? Can you see this is giving the structure of cylindrical? So your microtubules are nothing but which gives the structure, but it is going to be hollow and cylindrical. It is going to be hollow and cylindrical, right? So how many protofilaments are present in a microtubule? We have 13 protofilaments. These 13 protofilaments will combinedly form one microtubule. Can you see how these 13 filaments are arranged like this circular one? Beech mein hollow will be there, and it gives the appearance of cylinder. This is going to form your A microtubule, which is the complete microtubule. But when we have 10 protofilament instead of 13 protofilament, it forms only and only one microtubule, and this type of microtubule we call it as, it is going to form another microtubule. This type of microtubule we call it as B microtubule, which is complete or incomplete? It is incomplete, right? So I hope you understood about the difference between A microtubule and B microtubule. So an exonym is made up of 2 central and 9 peripheral. This is what we have seen in the diagram. Each peripheral doublet consists of A microtubule, B microtubule. A microtubule is going to be smaller, present at the outer surface. It is complete, and it is going to have 13 protofilament. What about the B microtubule? They are larger than the A microtubule. They are inner surface, at present the inner surface, they are incomplete, and they are there will be 10 protofilament present over there, right? Now peripheral doublets are interconnected. We have seen the interconnection between them. I think I forgot to explain you that. Okay, so there is the interconnection between A and B. A is again stronger, so A is going to connect with the B of the other doublet through a protein. We call it as nexin. Nexin is the elastic protein. Now again, why elasticity, ma'am? Because just now I told you that the doublets are going to slide over each other, so that means there will be slight movement in the position, slight change in the position of doublet occur. So if the nexin is not elastic, what will happen? There will be the rupture in flagella, right? Now let's count it again, class. This is very important. Neat. With respect to the neat, this question was asked once upon a time. Arrangement was asked in the neat, right? At periphery, 9 doublets are there. 9 into 2, how many microtubules? We have 18 microtubules. At center, we have 2, that is A and A. So what will be the total number of microtubules present in flagella? We have 20 microtubules, right? Now, we have also studied about the dynein arms. That is nothing but A tubule, where dynein arms are present, it is present on the A tubules, right? Now A tubule faces the B tubule. So A tubule which faces the B tubule of the other one is going to have dynein arm like this, because it has to attach with the another doublet for the sliding movement, right? They are oriented in a clockwise direction. In this direction it will be oriented, the direction of microtubule. They are considered as a protein motor molecule. What is motor, class? Which gives energy. So dynein protein is the one which is going to give energy, because it has ATP synthesis. We'll study in the upcoming classes. They are made up of protein. We call it as dynein. They are made up of protein. We call it as dynein. Then what about inner sheath and outer sheath? Inner sheath, I have already told you that it is going to cover 2 singlets. They are fibrous in nature. Now just imagine if this is also elastic, your radial spoke is also elastic. So what will happen? All will be elastic, and it will be like a jelly dance movement, right? But we don't want that. Something has to be stick, right? So this fibrous one it's little stronger as compared to the elastic one, stiff as compared to the elastic one. Now the outer sheath is nothing but the extension. This plasma membrane is nothing but your outer sheath, and this surrounds the 9 doublets, right? The central singlets do not reach below the level of pellicle or plasma lemma. This I will be explaining you. Put question mark over here, right? Now I think, okay, let me explain this, then I will explain you that. Now, radial spoke, if I say, what is the another name of radial spoke? It is radial bridge. How many radial spokes are there? All the radial spokes comes from A microtubule. We have 9 A microtubules, so 9 radial spokes will be there. It is again made up of elastic fiber. It resembles, how does it looks like? It looks like the spokes that connects to the rim of the wheel. आपने अगर bicycle देखी हुई है, तो bicycle में it is going to be the tire. If you'll see, it is going to be like this, right? So it looks like that. This this as it has compared with the radial spoke, right? What is the role of it? It limits or control the extent of sliding. Now one doublet has to slide over the other doublet. If it slides more, then there will be the different kind of movement occur in the flagella. This is not good for the organism. So it controls the sliding movement of the doublets over each other during the bending movement of flagella. What is bending movement? We are going to study in the next class. Okay. Now, last one, that is basal granule, or which is also called as kinetosome or basal body or blepharoplast. These all names are super important. You're supposed to remember it. Now, where it is located? It is located in the ectoplasm below pellicule. Okay, now let me just explain you about pellicule and then we can move ahead. Now this is the picture I have taken from your textbook. Okay, here you can see above the pellicule or pellicle, whatever you say, above the pellicule or pellicle and below the pellicule. What is pellicule? Pellicule is nothing but the proteinaceous layer, protein layer which gives flexibility, and it is the outermost layer, it is the outermost layer. Okay, so pellicle is nothing but the protein layer which is going to give flexibility to the flagella, and it is going to be present at the outer layer, right? Probably you will study about this in your botany chapter, biological classification in euglena. Euglena does not have plasma membrane, it has a pellicule. Okay, now above, so this is going to be your pellicule. Now above the pellicule, by the way, why I was explaining it's okay for the pellicule, right? So this one is pellicule class. This one is the outer one, we call it as pellicule. Above the pellicule, the structure, there will be the structure of flagella, and below the pellicule there will be the structure of basal body. Okay, basal body. We'll come back to this slide again. Let me first explain you this. Okay, so basal body acts like a cell organelle involved in the formation of flagella and cilium. We have already seen. The basal granule, what is it? It is basically the modified centriole. Class, what is centriole? First understand that. Centriole is nothing but, like, if you'll see your cell, we have different types of cell organelles like mitochondria, ribosome, endoplasmic reticulum, Golgi, and etc. Same way, we have one cell organelle which we call it as centrosome. These are basically present only and only in animal cell, won't be present in microorganism, prokaryotes, or your plant cell, right? It is present only in the animal cell. They are membraneless. They do not have any membrane. Now, so this, if you'll consider this, we can consider as the centrosome. So centrosome has two cylindrical structure which we call it as centriole. This is your one whole centriole, and this is your, let me color it with another one. This is your second centriole. Second centriole. Now class, tell me, these centrioles are parallelly arranged or perpendicularly arranged? They are arranged perpendicular to each other, right? And these centrioles are helpful in the formation of spindle fiber. Now these spindle fibers are going to help in our cell division, right? So centrosome is nothing but the cell organelle. In the cell organelle, there will be the formation of centriole, right? Now, see, where is my pen? One second. Okay. Now, they are triplets. What we study about the flagella, they are doublets. They are doublets. But when we talk about the centriole, they are triplets. Let's see the structure of centriole. Again, this is very, very, very important. They ask question about the cartwheel structure. Who gives the cartwheel structure? Who resembles with the cartwheel structure? Don't get confused. Flagella is not going to be resembling the cartwheel structure. It's your centriole which is going to resemble the cartwheel structure. So if you will see here, we have A microtubule, B microtubule, and C microtubule is present over here, which was absent in the flagella. So A microtubule of one triplet is going to be in connection with the C microtubule of other triplet with the AC linker. Do we have any singlets in the middle? We do not have any singlets in the middle. We have proteinaceous hub. These proteinaceous hub are connected with the A because it is the complete one. It is going to be connected with the A microtubule by radial spoke. So class, here what is the arrangement? 9 triplets are there. So 9. Do we have any singlets in the middle? No. So 9 plus 0 is the arrangement present in the basal body or the centriole. A will have your 13 protofilament, B will have your 10 protofilament, and C will have your 10 protofilament, right? Now, see here, again the calculation at the periphery, we have 9 triplets, so 9 threes are 27. Do we have any microtubules in the center? No, we do not have. So the arrangement is going to be 9 plus 0, that is total 27. Now class, one centrosome mein how many centrioles are present? 2 centriole. One centriole will make your 27 microtubules. So this whole calculation, this whole structure is only for the one centriole. So 2 centriole will make 27 plus 27, that is your 54 microtubules, right? Now, whatever I have explained you on the diagram, same thing is given in your textbook. Listen to me very carefully. Before that, let me explain you one thing. Class, if you'll see the arrangement of A to C, if you'll see arrangement of A to C, A is in the center and C is towards the periphery. So, arrangement from A to C is from the center to the periphery, right? See here, the tubule of each triplets are designated A, B, C. So, tubules, these are the microtubules A, B, C. From center, that means A will be towards the center and then B, and C is towards the periphery. C is towards the periphery, right? Both A and B are going to cross the basal plate. Now listen to me very carefully, class. Just now I told you that the basal body is going to form your flagella, right? But in flagella, what we have seen only A and B. There is no C. So here, see here, this is the diagram. So this one is your basal body and these are your microtubules. Can you see? Parallelly they are arranged. So these are the microtubules of A, B and C. As soon as it reaches the pellicle, what happened? The C gets terminate over there. Their growth stops over there. Only A and B are going to extend beyond the pellicle. What about the C? C is going to terminate over there. So only A and B are the one who is going to be present in the flagella, but A, B, C, all three are going to be present in the centriole or the basal body. That's what is written. Both A and B cross the basal plate. This is your basal plate that is above the pellicle, and continue as the peripheral doublets above the pellicle in the exonym. But what about C? C gets terminate at the basal plate, right? Thus, triplets are the basal granules thus the triplets of the basal granules are converted into flagella because it got converted into, it is only having A and B microtubules. So the basal granule which had A, B and C got converted into flagella and ciliary which has only A and B. There is no central microtubule present in the basal granule. The basal granule is connected to the plasma membrane and nucleus. See here the structure of euglena. This is the structure of euglena. Now this is going to be your basal body, and from the basal body your flagella will come. There will be small flagella and there will be the large flagella. Two flagella will be there. So here you can see the nucleus over here. The basal body is connected to the plasma membrane. Here you can see plasma membrane connected to the plasma membrane and connected to the nucleus through what? Through a communicating structure. We call it as rootlet. So rootlet is nothing but your communicating tubule. That means basal body is going to communicate with the plasma membrane as well as with the nucleus through the structure. We call it as rootlet. Rootlet are the one which is responsible for pulling the flagella, and also it gives the orientation, that in which way it has to move, right? Now, so later about the lateral appendages we are going to study in tomorrow's class, because there are the types of flagella we'll be doing with the lateral appendages, right? So I hope this must be clear to you, class. I think I did not explain you much about the rootlet in the class. So please understand, rootlet is nothing but the communicating tubule. Like we have A tubule, B tubule, same way we have another tubule that we call it as communicating tubule. So it is going to be in connection with the plasma membrane as well as in connection with the nucleus, and this helps in pulling the flagellum. This flagella is being pulled by using rootlets, and it decided in which orientation, in which way it has to move, right? So that's all we have in today's class. I hope you understood today's topic. It is little different, little difficult. Please go through the video again and again, make your notes and understand the concept. Thank you everyone.